Now let's take a look at an example immune response. This will give you a basic idea of how our immune system reacts to infection, and then we'll build on this knowledge using infections we vaccinate against as examples. Let's base our example on community-acquired pneumonia. It'll be caused by a bacteria or agent that we've never previously have been exposed to. First, we'll look at the primary immune response, and then look at what happens when our body encounters the same pathogen again. As far as our pathogen goes, it's not important at the moment. It's a bacteria and it has some antigens that our immune system can attack. That's all we really need for now. Source of infection. In actual clinical practice, it's rare to identify a source of community-acquired pneumonia. There are some cases where water sources contaminated with Legionella cause outbreaks, but this is beside the point. For our example, let's assume you've come in contact with someone sick and they sneezed in your general direction a couple of times. Route of transmission, airborne. As is common for most respiratory infections, they are transmitted through the air that we breathe. Our respiratory tract has some defensive measures in place to make sure we won't be infected. After all, if we didn't have any, we'd be getting pneumonias all the time. Most of those are non-specific, and I'll mention a specific one when we get to our secondary response. For now, let's stick to our example and assume that our hypothetical patient contacted a large enough dose of the pathogen. It resulted in bacteria breaching through our defenses and being able to reach the bronchi and alveoli. Here's a rough graphic representation of how our lungs look like under the microscope. The giant holes represent the alveoli. They're the most terminal part of our respiratory tract, and it's the place where oxygen can migrate through and enter our bloodstream. You can also see parts of other bronchi that lead to other alveoli, and also some blood vessels. Now the bacteria have found a place to settle, and they're going to multiply. This is the gist of the incubation phase. The bacteria grow and multiply, but our bacteria can't produce resources out of nowhere. In order to get enough food, it destroys the cells around it, the cells of our lungs. Clinically, this phase has no symptoms. There isn't enough bacteria at the moment to cause significant harm, and our body can compensate minor damage very well. In fact, most of our organs are pretty good at working overtime. You can completely remove one lung from a person and they'll never feel a difference unless they're into sports. Our liver fails if at least 85% of it is damaged and can still make a full recovery afterwards. Symptoms of chronic kidney disease occur only when kidneys lose over 90% of its functional capacity. But I digress, let's get back on track. Bacteria keep damaging the tissues around them. Cells that are killed off release certain chemicals called attractants. These chemicals attract granulocytes. Remember, granulocytes are responsible for inflammation and non-specific immune responses. They're able to kill off some of the bacteria as well as initiate inflammation where bacteria resides. Same chemicals, as well as the ones released by granulocytes, also attract macrophages, natural killers, aka NK cells, and the complement system. Macrophages engage in a process known as phagocytosis, and all cells capable of it are known as phagocytes. Phagocytosis is a process of eating another cell. After being eaten, the bacteria is exposed to a bunch of aggressive proteins designed to kill it. Most of the bacteria is indeed killed off by that process. NK cells are a type of lymphocyte that have some generic tools up their sleeve. They're trained to fight off against some common antigens and do a decent job of it. The complement system is a collection of proteins. They form a cascade activating one another until one large protein, known as membrane attacking complex, forms. As the name implies, it's a protein designed to damage a bacteria's membrane. Membrane is arguably one of the most important components in every cell, and if it doesn't function properly, the cell dies. In a lot of cases, those mechanisms are enough. Our body deals with millions of bacteria on a daily basis, and we don't get killed off by them thanks to these defensive measures. But in other cases, they're also not enough. Eventually, the bacteria manage to increase their population and break through. As the incubation period ends, there is now too much bacteria and not enough food for them. They start damaging the lungs even harder. The inflammation caused by granulocytes also plays a role, as those parts of the lungs function much worse now. Our patient starts showing symptoms of a respiratory infection. Weakness, fever, cough. as well as difficulty breathing. So let's talk about the mechanisms of specific immunity. Mind that, they kick off right at the same time as the non-specific ones. Problem being, they are rather slow and also rely on previously mentioned mechanisms. Specific immune response starts with antigen-presenting cells. These are phagocytes in nature that consume bacteria. The bacteria is then killed off, and only some of their toxins remain. Toxins are then further broken down into antigens, and antigens are placed on top of APC's receptors. APCs then migrate to primary and secondary organs of the immune system where T helpers reside. T helpers are then presented with antigens. T helpers, as you may remember, are responsible for both humoral and cell-mediated immunity. Cell-mediated is a bit more straightforward. T helpers start screening for lymphocyte precursors that can develop immunity for a specific antigen. Then, they screen the same lymphocytes a second time, making sure none of them can target our body's own antigens. This process is known as double selection, and lymphocytes that successfully pass it are then given a command to multiply. The result of cell-mediated response is the formation of T killers. These are lymphocytes that can target a specific antigen. They are then released into the wild to deal with the bacteria. Humoral response is a little more complex. At first, it follows the same principles. APCs consume the pathogen, kill it off, and present antigens to T helpers. T helpers then start screening for B cells that can attack the pathogen. B cells that successfully undergo double selection are then given a command to multiply. Once they multiply, B cells turn into plasmacytes and start producing antibodies. Remember that the default mode of B cells is the production of immunoglobulin type M. These antibodies are relatively specific, and it's our immune system's more immediate way of reacting to pathogens. For most infections, it takes about a week to form IgMs, and their production peaks another week afterwards. Then, they disappear just as quickly. IgMs are a decent solution to the infection, but there are also a few problems with them. First, they are much bigger and therefore take a lot more resources to produce. Their peak plasma concentrations may never reach appropriate levels to defend against a pathogen. 2, they are only relatively specific. And 3, they don't have a lot of cellular binding sites. But they do their job as they greatly amplify the non-specific responses and allow for more specific ones to form. As our immune system starts to fight properly, APCs are also able to present much higher quality antigens. On top of it, with more time, our T helpers are able to screen for better responses. This kicks our best line of defense against infection, the production of immunoglobulin type G. B cells undergo the very same process as before, except they're also given more time and care to become a different type of plasmacyte, one that can produce IgGs. IgGs are better in a number of ways. First, they're smaller and thus don't require too many resources to produce. Their size also allows for other certain benefits, such as being able to cover the bacteria more efficiently and migrate through the placenta as well as into breast milk. 2, they're highly specific. And 3, their production isn't limited. Their peaks can be very high, and they can potentially remain in someone's blood for the rest of their lives, provided protection against reinfection. IgGs generally start appearing a week after IgMs reach their peak. Their own peak is reached after another week, and it takes a very long time before they disappear. Antibodies act in a variety of ways when it comes to preventing infection. First, they can simply cover and neutralize important bacterial antigens. Our strategy for vaccination against hepatitis B does exactly that. Second, antibodies can attract all kinds of agents: granulocytes, phagocytes, lymphocytes, as well as some proteins of the complement system cascade. Antibodies are not only effective on their own, but they also help amplify other types of responses. Generally, the formation of antibodies is associated with the resolution of clinical symptoms. IgMs may not be enough to deal with an infection, but the picture starts to look much better after IgGs are produced. In our case, it takes about a week for the person to start feeling better, and a few more weeks until the full resolution of their symptoms. Now, our body doesn't just fight off a pathogen and gets done with it. It prepares for an incoming invasion yet again, and there are two ways of defending that I quickly want to discuss. First, the formation of memory cells. In both humoral and cell-mediated responses, memory cells are formed when lymphocytes are commanded to multiply. These cells stay with us for a very long time, and if they're able to recognize an antigen, then they instantly start to multiply. Second is the formation of another class of antibodies, IgA. These serve a variety of functions, but most notably they are referred to as barrier antibodies. IgAs, once produced, migrate and reside in tissues that contact with the outside world. If a pathogen they recognize comes in contact again, they're quickly able to neutralize them and amplify our non-specific defenses. Now with this in mind, let's briefly take a look at secondary immune response. Our patient has contacted the same pathogen again. The source of infection is the same, the route of transmission is the same. However, now the bacteria has to face a few problems. First, IgAs are now a threat to them, quickly preventing them from being able to effectively grow. Second, we could already have IgGs and T killers from the last time we got sick. In this case, there's no need for any further response. Our body can already fight the bacteria off. In case none of those defensive mechanisms worked thus far, the same stages as before take place. Except this time, memory cells also kick in. Once an APC presents them with an antigen, they're swiftly given a command to multiply. The new cells quickly turn into T killers and plasmacytes that produce IgGs. As a result, highly specific immune response can kick in right off the bat. Afterwards, all of the same mechanisms of immunity take place. As far as antibodies, both IgMs and IgGs are produced and peak about a week after the incubation period. IgMs are produced in a relatively low amount though. There are a few ways secondary interactions with the pathogen are different. First, there may not be any infection at all. Having antibodies means that one may never have to worry about a certain pathogen ever again. This exact idea is the basis of vaccination. Being immune to a pathogen means no infection for the most part. Another desirable outcome is the lighter clinical forms. In tuberculosis, for example, vaccination isn't able to entirely prevent the disease, but it's been observed that people who get vaccinated don't develop the most severe clinical forms of the disease. Anyways, that's how our immune system reacts to infectious agents. I hope the visual representation has been somewhat helpful in forming some understanding of how the process actually works. Next up, we'll discuss a few basic ideas about vaccination.